Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 159 for the podcast. This week's episode is brought to you by MedChem Express. MedChem Express is a global supplier of research chemicals and bioactive compounds, offering more than 150,000 bioactive molecules. With worldwide warehouses, MCE ensures fast delivery and stable pricing. Their portfolio includes inhibitors, agonists, labeled compounds, proteins, peptides, antibodies, dyes, and screening libraries, supporting drug discovery from start to finish. Supported by a team of seasoned professionals, they are committed to delivering friendly and proficient service to cater to your unique requirements. Visit medchemexpress.com to start your research journey. Recently, the UK government announced plans to sequence the DNA of every baby born in that country. A part of the country's 10-year health plan, the program aims to identify rare genetic diseases early, prevent harms from delayed treatment, and reduce future healthcare costs for the NHS. Yet along with those potential upsides come a range of ethical and practical questions too. Who guards the sequenced data? Who can access it? How can consent be informed, managed, and withdrawn years later? And what is the best way to gather the genetic material required to fully sequence the DNA of 700,000 infants each year? This week, I spoke to Neil Ward of PacBio to understand more about this program, compare it to other national genome sequencing programs in Europe and Asia, and discuss the promise of unlocking the language of life. I hope you enjoy my talk with Neil Ward of PacBio. Neil Ward, welcome to Beyond Biotech.
Hello, nice to see you again, Dylan.
Neil, give me a little bit of your journey through the biotech industry. You worked in genomics, you've worked at companies like Agilent and Illumia, to your current position now at PacBio. What's been the story there?
Yeah, hi, Dylan. I guess it's a bit of a long, winding journey, but I did my degree in biochemistry. I decided I wasn't very good in the lab. Did a follow-up master's degree in bioinformatics. I decided I was more comfortable with computers than I was pipettes. And that was really the start of my sort of educational journey. And from there, I've worked in various different companies in the biotech sector. Initially in research, sort of bioinformatics for a couple of small biotech companies in the UK, and eventually transitioned from working on that side of the research environment into many of the companies that supply tools, equipment, software that sells into the pharmaceutical and or the research setting, which can be academia as well. And so today I find myself as VP General Manager for PacBio. So we're a Californian-based company. We are one of a number of companies in the world, there's probably half a dozen, that make equipment for looking at DNA sequences. And that takes all sorts of different forms. It's a fantastic, interesting industry as a whole. DNA touches all sorts of things from human health all the way through kind of plant and agriculture, is how we can grow crops that might be drought resistant, for example, or I'm sure many of the audience are familiar. We also played a role during COVID of looking at the sequences of the virus as it was evolving. So I think there are a number of other companies that I've worked for previously as well. And it's an exciting dynamic space at the moment. There's a lot of competition between the various players, between Pacific Biosciences and other companies in the area, which I think is great. Keeps us on our toes, keeps us busy and innovating, and ultimately it's great for the researchers. We continue to drive down the costs, increase the throughputs, and start to enable science with genomics at a much bigger scale than previously.
So you're a VP there at PacBio. What do you actually do in that role in Europe and Middle East and Africa? And where do you see yourself actually contributing to the future of genomic healthcare in the region?
My team would probably ask the same thing, Neil, what do you do as VP? But I've got a great organization. We have an office in London, so near King's Cross, which is a really nice sort of dynamic part of the city and a growing tech hub as well. So it's got some great neighbours that are biotech companies and the big likes of Google and others in that space. And then as a team, I lead a small sales team that are based often in home offices, but travel around and meet our customers and try and advocate for our technology. And then we have a small group of marketeers that arrange a lot of the trade shows and a lot of the events that we try and put on to educate customers about the value of our technology versus others in the marketplace. And then we've an amazing group of engineers, folks that get into the guts of the instrument, do the installs, understand everything from the compute hardware to the lasers and optics and stuff that is associated with our technology. So really a broad group of individuals. There's just over 40 people in our European team that are going out there selling, supporting, and ultimately helping customers around Europe, Middle East, and Africa take our technology and do some amazing science with it.
As well as these individual clients, so you've also been involved in some pretty major national genomics programs, things like the Genomic England's 100,000 Genome Project, the UK Biobank. How are these sort of lessons shaping what you're doing and also what your team's doing?
Yeah, I've been, in some respects, fortunate to be at the right place at the right time during my career. So I'm not, some of it's luck and some of it's skill, and there's probably more in my case of luck. But from a timing perspective, I spent a long time at Illumina, which has been really the dominant player in this market for a long period of time. I joined there in 2008, just as the American organization acquired what was a spin-out from Cambridge University in the UK called Solexa at that point in time. So I was one of the first commercial members of that team, and that went from a very, very small company to a multi-billion-dollar industry over the course of that following sort of 15 years. And that gave me a unique opportunity to shape the direction of genomics, interact with many thought leaders, both in the academic and the political space, in pharma as well, and really, brainstorm what is possible, and I think over that trajectory, the cost of DNA sequencing has declined precipitously. So 20 years ago, it was $200,000 to do one genome. It would take weeks to do the sequencing, weeks more to do the analysis. And that, as of today now, is just a matter of a few $100s and sort of 24 hours. And that opens up all sorts of possibilities for how genomic information can benefit human health. And so, yes, that's taken the form of lots of big projects. I think the UK government has been really leading the way globally. All governments around the world like to say they're world leaders, but I do think genuinely the UK, with projects like Genomics England, are clear leaders in taking that sequencing technology and applying that in a healthcare setting. It was the first country to offer whole human genome sequencing for individuals with suspected rare disease. So these are typically children that are missing their developmental milestones. There's something that looks not quite right. Both parents are typically healthy, but a child has just been really unfortunate and had a spelling mistake in their genetic code in one of the 3 billion letters that encode your DNA. And whole genome sequencing has allowed many, many of those patients now to find the underlying genetic cause. Sadly, in many cases, that doesn't result in a drug or a change in prescription, but it often is really informative for the families. They can find other members and build a community of people that are going through that difficult journey with them. And in a small number of cases, it has been transformative, sort of, just either modulation in just some of the vitamins that maybe they're deficient in can actually significantly reduce the deleterious effects of their disease if caught early. And so that's been amazing to be part of that. And we're seeing now the technology moving into cancer care, into preventative medicine as a whole. And so, as I say, I've been fortunate to interact with many of the thought leaders in the world and help shape a little bit of that conversation.
PacBio has been around for a while now, but genomics itself has changed enormously in the last 10 years. You've mentioned, obviously, the price and the speed as being two areas where it's changed, but how has your company actually changed in that last decade or so?
Yeah, as I say, PacBio has been around for 20 years. It's been a company that's always had some fantastic science at the core of its technology. But I think internally we'd acknowledge that historically, while the technology was amazing, we maybe weren't commercially at the right price points and throughputs. So I mentioned earlier, Illumina has been really a market leader in this space because they were able to take their sequencing methodology and parallelize it so that they can take your human genomes, your big chunks of DNA, smash that up into tiny, tiny little pieces, and then do billions of reactions on billions of fragments and sequence each of those little pieces of DNA and stitch together with computers that sequence and align it back to a reference genome. That's a very, very powerful way of doing the sequencing and gives significant cost advantages. This is why they've been able to drive down the cost of sequencing as computing resources have improved, as the ability to basically look at more fragments of DNA down a microscope over time has improved. Where PacBio has been different is that instead of smashing genomes up into typically a couple of hundred base pairs, we try and maintain the structure and the sequence information into much larger fragments, with the overall belief that it's a little bit like a human jigsaw. You would much rather do a child's jigsaw of a few pieces that are easy to stitch together and know where they belong than one that is billions of pieces where you've got lots that are blue and you're like, I don't know, it's sea or it's sky, or it will go roughly there, but you don't have that precision. And we genuinely believe that precision that we have with the more accurate assembly of a genome will allow us to get accurate information from the bits of the genome that are difficult, that vary a lot between one individual and another. And that's part of what motivated my move to actually move from Illumina to PacBio. I could see that the PacBio's technology was starting to evolve, starting to scale more effectively. And I think over time will become a serious challenger to Illumina with regards to getting similar price points, but with a much higher fidelity product.
You're based in London and you're focused on the EMEA region. Where do you see actually, I guess, space or areas for growth in that region? Where can PacBio really start to take advantage of the region commercially? Where are you going to see the growth?
Yeah, so at the moment, I think short-read sequencing technology plays in lots of different applications and market spaces. But where our sweet spot is at the moment is we have this combination of very accurate sequencing with these long reads that give us a very complete picture of a genome. And it is actually this rare disease space that I touched on already in the interview that is our sweet spot, because you need to find the 1 in a 3 billion base pair changes. And you don't want to rely on just accurately sequencing the bits of the genome that are easy to sequence. Some of those changes occur in your X chromosome or your Y chromosome, those chromosomes that confer our gender. Actually, they're very difficult to analyze with typical short-read sequencing because they're very repetitive. They've got lots of bits of DNA that look like one another, lots of repeats in structure and size. And so we've got some fantastic publications and collaborations with big academic medical centers across Europe. There's a group in the Netherlands and a place called Radboud in Nijmegen who've done some amazing work and presented a lot of their work at the European Society of Human Genetics earlier this year, starting to show that the PacBio sequencing in that rare disease setting is really the only single genetic test that can replace the multitude of tests that have gone before. If you go into any human clinical genetics lab, what you'll find over the last 20, 30, 40 years is that they've had a technology, it's had a certain characteristics, and then a new technology has come along and they've added that new technology. But in many cases, they've still needed to use the old technology. So they're still using pulse-drilled gel electrophoresis on top of chromosomal microarrays, on top of FISH probes, on top of MLPA, on top of exomes and genome sequencing. It's just been this one after another. And so the promise of genomics, of decreasing that cost of the genome, hasn't really materialized as much as it could do, because you've still got a lot of these legacy tests. And what the Radboud team and now others around Europe have shown is that a hi-fi genome, because it can get into all of those difficult bits of the genome, can actually replace all of the multitude of tests that have gone before. So while our sequencing of a single genome may be still slightly more expensive than the short-read technology, you get that cost savings and efficiency savings of being able to finally move away from all of the old legacy tests and have a single test that's a universal test for all patients coming through. And they're scaling now to thousands of genomes there in the Netherlands on an annual basis for anyone that presents with a suspected rare disease or a genetic variant that's suspected to be germline. The same is true in Sweden, in Denmark. But you often find the rates of adoptions per country are influenced by all sorts of things, not just the technology, but political will and the state of the healthcare system, the funding that's available. We've maybe not made as much progress yet in the UK as a company. which is a personal frustration for mine, being an Englishman living in the UK. But I think there's a lot of opportunities in that space. Outside of Europe, where maybe we think about healthcare more often, there's a huge genetic disease burden in the Middle East. You may not recognize this, but in many countries around the world, if you take the UAE, or you take Qatar, Bahrain, Saudi Arabia, they have very high rates of first and second cousin marriages. Historically, they wanted to keep wealth within families, and that means over generations they've been marrying various cousins, and as a result, you do see a much higher rate of what would otherwise be rare diseases in normal populations. So we've done quite a bit of work out there. We've had some publications out just recently from collaborators at Mohammed bin Rashid University, which is based in Dubai, where they've been able to use our technology to create what are called pan-genomes. These are, instead of aligning your DNA sequences to a single linear reference genome, they try and aggregate multiple completed genomes that go from one end of a telomere to the other end in multiple individuals, and then make like a tube map of all of the different genetic variations and how they vary from one to another. So that was out in Nature Communications just in the last couple of weeks, and they found over 100 million base pairs of new DNA sequence in those, I think, 47 individuals that they assayed in that small study that weren't in the reference genome. And the exciting aspect of that is we see that around the world. We see that in the Middle East, but we also see that over in Asia, in Africa in particular. Millions of base pairs of DNA that aren't in the reference genome. Now, with the technology that we bring to bear, we can actually start to ask the questions, well, in that population, do any of the bits of DNA that are novel in those ethnic minorities affect their health? Before, without a reference genome to align those fragments of DNA, they ended up in a bin of bits that didn't seem to fit the jigsaw. It's a bit like when you do your IKEA furniture, there's the screws left over afterwards. Those were the bits that were left over afterwards, and you kind of go, oh, well, I'll put them in a drawer, I'll worry about those later, and nobody ever goes back and looks at them. But now we have the means and tools to do that.
Neil, you mentioned a couple of minutes ago the UK was really standing out as a leader in sequencing, particularly for rare disease. But it's a recent announcement that they made that really gave me the impetus to reach out and talk to you, which was their plan to sequence the DNA not just of certain children, but of every baby born in England over the next decade. Why is this such a significant step, and how does it align with what other countries are doing for newborn screening?
Yeah, again, I think this is where the UK are leading the way, and this has been a project where Genomics England has been leading this in conjunction with the NHS, and they've been really thoughtful, which I think is important in this situation, on how to do this. They've done a lot of consultation work with expectant families and the population as a whole. And I think here in the UK, at least, it seems that the general population is ready and comfortable to allow this to happen, and trusts the NHS and Genomics England to use that data in the right way. And those are really, really important aspects of any of these types of projects. But the hope and the ultimate aim is that if we can sequence an individual's genome at birth, or at least very close to birth, it typically would take some cord blood during that process, do the sequencing over a few days maybe, and then that data, the genome, is part of their medical record for life. And I think that doesn't mean necessarily from day one you start to try and work out whether they're going to have Alzheimer's when they're 70. That's probably not something that they would do at that stage. But it's a resource that you can go back to at any point in time when other things happen, other phenotypes start to appear. It's a reference data set that can, I think, hopefully really inform treatment and hopefully start to move the NHS towards more preventative healthcare. I think what it will do is it will circumvent the need for rapid sequencing for children with rare genetic disease. It's a very small percentage of the population, but at the moment there is that challenge. It's typically, a child is born, it looks fit and well and healthy from day one. The parents take it home, and it's often a few weeks later, maybe they're failing to thrive in some way, or maybe even a few years later, where their intellectual kind of development isn't occurring as one would expect. They're missing some of their developmental milestones. And then we need to get this answer really quickly. It can often be an acute setting. The child is presented with maybe some vitamin deficiency and they're not thriving. Instead of rushing to then do the genetic analysis and taking blood samples, at that point in time, you could just refer back to the electronic medical record, back to the DNA sequence that was generated at birth, and hopefully have a quicker intervention. Or if the process is really kind of done at scale, maybe that was identified proactively at birth, and maybe that child didn't even present with those conditions because you've been able to put some interventions in place that have helped. So I think there's huge possibilities of how that data can get used over a lifetime. And as the costs have come down to actually generate that data, it almost seems criminal not to do that now. We're talking, certainly with all of the costs rolled in, under £1,000 to have your genome as part of your medical records for life. And that genome can be used in so many different ways through someone's life course that I think it'll easily pay for itself with regards to the overall healthcare economics of it. But it's a journey that we're on. The UK is leading the way with the generation study. They're starting that with short read sequencing, but they're actually undergoing a consultation process with industry and partners at the moment, and they're going to do a test to see whether long read sequencing technologies actually add additional benefit over short reads, and how they'd trade the various pros and cons off between the technologies as well. So a really exciting time for implementing newborn screening. And I think the UK is just one of a number of countries that's thinking along those lines.
There are practical challenges to doing this. There's also some ethical challenges. You mentioned there things like having a high trust society that trusts the NHS not to do something inappropriate with the data. There's parental consent that you've got to do. How can you actually address those sorts of concerns, the ethics and the practical side?
Yeah, it's probably a little out of my personal area of expertise, but the project as a whole has had a significant working group on there that has engaged multiple stakeholders in opinions on that. The security is going to be essential, making sure that is a data resource that is, I'm reluctant to say impossible to hack. I don't think any systems are ever impossible to hack, but at least is very, very secure. Because genetics data does concern people in different ways from other medical records. You can argue whether rightly or wrongly. But one thing that is for sure is if you have my genetic sequence, you've been able to get hold of that, you'll be able to tell something about my risk of diseases. But not only that, you'll be able to tell something about the risk of diseases that my siblings will have, that my parents have, that my children have as well. And so not only do you have that sort of individual consent aspect of genetics, you kind of have the wider consent of the family as well. And that's the bit in genetics that does get a little bit tricky now. And that's often why people get so concerned about, well, say you've got a twin. This is the kind of ethical bit that gets a really little bit weird. One twin and their identical twin share almost identical DNA. Now, one of those two twins can independently agree and consent to have their whole genome sequence on the internet and stick it on Amazon and make it publicly available for everyone. What rights does their sibling have? Their twin whose DNA is almost exactly the same. They may have one or two errors or differences between them. So there's a lot of interesting ethical conundrums that do roll into this space. And then, especially in the newborns area, that's not the individual that is consenting, it's their parents at birth. So all the processes have got to be in place that individual, as they age and become, inform themselves, they can opt out or opt out really at any other stage, at a later stage in life. We're fortunate in the UK, we have the NHS, so there's less of a concern about this information being used against someone to price insurance and those sort of things. We've got, fortunately, an NHS system that gives us access to care for at the point of care. But there is also a moratorium amongst the insurance industry that it won't use genetic information against someone if they're trying to take out a life insurance policy or other kind of care. So I think we're relatively well set up, but time will tell on how that program goes, and I'm sure there will be ethical conundrums that haven't been thought of during the consultation processes that come up as we go, and one would hope that those are treated carefully as we go through the process.
You've mentioned a couple of different approaches to sequencing, the short read and the long read. Can you tell me what's the essential difference there, and why is the long read sequencing better suited to a project like this?
Yeah, so both technologies ultimately, and there are multiple different vendors now that do short read sequencing technology. Illumina has been the larger player, but there's a growing number of competitors in that space, and that's where they take DNA, cut it up into tiny pieces, they amplify those, and then sequence with fluorescent nucleotides. So it's basically a glass slide with lots of bits of DNA stuck on it and a very high powerful microscope that looks at the little color changes as you incorporate a fluorescent nucleotide to build out a complementary strand. And that's been a very cost-effective way of doing genome sequencing, and many of the short-read sequencing providers now are in the couple of £100 to do a human genome. And then in the last couple of decades, there have been two companies, PacBio and Oxford Nanopore, that have been looking to bring to bear long-read sequencing technologies, with the overall hypothesis being that this fragmenting bits into tiny pieces actually makes it really hard to do the assembly to find where the differences are. But historically, the longer DNA sequences have been less accurate in their fidelity. They've typically been also significantly more expensive and lower throughput. You can't do as many samples. But I think that's where both long-read sequencing technology companies have made significant innovations over the last probably 5 years in that time frame. So here at PacBio, we have a sequencing platform that we call the Revio now, that's our market-leading platform. That can now do 2,500 genomes in a year. And our list price for a human genome is just under $500. So we're still more expensive than the short-read sequencing. We can't sequence as many genomes as the long-read sequencing platforms do per instrument. But that gap is now significantly smaller than it was even just a few years ago, where our older innovations, our older platforms, the Sequel instrument had 15 times less throughput. And so at that scale, it just wasn't possible to even consider doing populations or newborns. It's still challenging. We need a lot of our instruments in a room to be able to do that. But I think we're continuing to see a declining cost of the long-read sequencing and a slightly shallower declining cost of the short-read sequencing, such that maybe they won't exactly converge, I think, but all of those costs are driving down at different paces. And I think there's a growing interest amongst researchers and clinicians to use long-read sequencing because it gives you clarity of your genome in certain particular important parts. And so one example is there's a gene called CYP2D6. Lots of genes have horrible names. I don't need to worry too much about the detail, but that one gene confers your ability to metabolize almost 50% of all pharmaceutical drugs. So if you take codeine, when you absorb codeine, that's metabolized by your body and hopefully converted into the active drug. Now, across the human population around the world, some people have 2 copies of that gene, but there's another bit of DNA just downstream that's called CYP2D7 that's almost identical in sequence. So if you're smashing your genome up into tiny little fragments, it's a bit like that jigsaw challenge where you've got a bit of sky and a bit of sea. It looks blue and you're like, well, I don't know, does it align to CYP2D6 or CYP2D7? And there's some ambiguities and uncertainties within there. And that means any guidance that can be given on the genomic sequencing that's been done with short reads may not have the precision that one needs to actually determine, do you need a half dose of that drug or do you need twice the dose of that drug? By breaking it into a smaller number of pieces, these large fragments, we can actually get both of those genes in the one fragment and understand, do I have the 2 copies of CYP2D6 that I should do and the 2 copies of CYP2D7? Or do I have some of the weird hybrids and crossovers and rearrangements that occur in that genomic region, which we're starting to characterize much more effectively now that we've got short-read sequencing? So I think there are examples like that where not every bit of the genome is equally important to assay accurately. There are some bits that are boring and probably don't confer much risk to disease. Other bits that are really interesting biologically, but are often difficult to sequence. And so I think increasingly we're getting more and more examples of the latter, where there are parts of the genome, the X and Y chromosome I mentioned earlier, that are highly repetitive. We've only just solved those and been able to actually even understand the sequence of it through a combination of our long-read sequencing technology. And I'm sure the X and Y chromosome confer some interesting biology. They define our sexes. So there's probably a lot of hidden associations to disease in these bits of the genome that are repetitive. And more than half of our human genome is made up of repetitive sequence. We underestimate that. But biology typically occurs by taking a bit of DNA, and if it's useful, it copies it and makes another copy. And then evolution occurs on that, and those things change very slightly, and then maybe it copies it again. So the bits that make us human, the bits that give us our characteristics and traits, are often bits of the genome that are made of these repetitive regions. And our eyesight, clearly something we rely on a lot as a species. There's a lot of repetitive genes, the opsin genes, that are almost impossible to stitch together with short read sequencing. Again, we can define those very effectively with our long read sequencing. So we're still scratching the surface, still a lot of biology to understand, basic biology to understand, but we think that ultimately there'll be a lot of additional value in there.
I guess if we're talking short read versus long read, we can also talk about where we get those samples from, because there are a couple of different places you can take it from. Traditionally, it's been the dried blood spots. We can also take it from cord blood for newborns.
Yeah.
Are there advantages in using one method over the other?
Yeah, you're right. So anyone that's had a child will at some point in time around the world have had a healthcare practitioner come to their house, or maybe do this in the hospital, and stick a little needle in their child's foot, and the child cries, and it's all very distressing for everyone as a new parent. And they dab that tiny little bit of blood onto a bit of paper, a filter card that they're called. And that's historically how newborn screening had worked. Those little cards of paper with multiple little blood spots that were spotted onto those would go to a biochemical lab, and then certain chemical tests, very cheap chemical tests, would be done on those blood spots. And different countries around the world have had different numbers of tests that they do on those dry blood spots, depending on all sorts of health economic situations. And the UK has had a relatively limited number of biochemical tests that it confers, that it looks for specific rare diseases. And the obvious thing is you can take that dry blood spot, and instead of doing a small number of biochemical tests, you can take that blood and try and do whole genome sequencing on it. Now, the challenge with the blood that's spotted onto a bit of card is, as soon as you spot it onto that card, it starts to degrade a little bit. It's not the most stable of way of keeping the DNA. And so while you can take a dry blood spot and do DNA sequencing on it, you typically have to amplify that material a little bit. And whenever we start to do amplification, we add some errors, some bias into the process. But I think all of the sequencing technology providers now have an ability to take one or a small number of those dry blood spots, amplify the material, and then generate a whole genome from that. And we can certainly do that. But it's not as pristine as it would do if you were doing it from fresh blood. So what the Genomics England study has done already in advance of the major rollout is it's looked at different ways of gathering the required sample for testing that newborn child, and actually recognized that there are different ways of doing it. You could take a cheek swab of that child as they're born, and that works. You can get good quality DNA from the cheek swab, but as you can imagine, if that child has just been born, it may still have some contamination from the mother in its mouth. And so that can then get very tricky from a DNA sequencing perspective. Are you sequencing the child or the mother, or how much of the mother, and bacteria and all sorts of other things? So it's possible to do that, but it won't be that process. What they've recognized, actually, the purest form of DNA that one can gather at scale and easily is cord blood. It's non-invasive. You don't have that crying child with the heel prick. You can just take a sample of the cord blood. You can get plenty of material such that you don't then need to do any amplification on it. So the Genomics England generation study is going to do that from cord blood going forwards, and I think that's a sensible approach. The other advantage of doing it from cord blood with our technology or long read sequencing technology as a whole is if you don't have to do amplification, not only can you sequence the nucleotides, your A, T, C, and G bases in your 3 billion base pair genome, but you can also look at some of the chemical modifications that occur in DNA, things called epigenetics. So typically, we also have this extra level of control of our DNA that's different forms of methylation, the most common of which is methylation on CpG sites. So a C next to a G in the human genome, the base gets methylated. You get a 5-methyl cytosine base. And with our sequencing technology, we can actually read that out. If we don't do amplification, we can read out the methylation changes in the DNA. So there's an additional whole area of research that we can then start to do of how is the epigenome of a newborn different from the epigenome of an adult. And we know from other studies that have occurred around the world that your epigenetic profile does change. It changes as you age. So there are specific parts of your genome that get more methylated as you become older, some that demethylate as you become older. There are parts of your genome that if you were a smoker, there's an epigenetic change that's characteristic of that individual having smoked or continuing to smoke. And so having not just the DNA sequence, but a kind of pristine epigenome at birth from these children in the generation study is going to be a really interesting resource to understand how epigenetics changes over a lifetime, how that changes over environmental exposures to pollutants and other things as well. So there's, again, another whole angle of research and a lot of scientists excited about being able to get hold of that type of data at scale.
You mentioned that the UK is really a leader here, but they're not the only government doing these newborn screenings. You're also
Correct.
collaborating with the Thai government. They have a national newborn screening program. How is the work that you're doing in Thailand, is that a model for other countries? Is it out there ahead of everybody else?
Yeah, so I think once you get out of the UK or Europe as a whole, there's even more need for genomics to happen. Unfortunately, as of today at least, there's still a real bias in the knowledge that we have about genetics, because the vast majority of genetics research studies that have occurred around the world have been in the UK or in people of European descent, either both here in Europe or in North America. And that's great. There's great studies like UK Biobank is a prime example. 500,000 healthy individuals gave their sort of medical records, blood samples, other phenotypic measurements to the global scientific society, such that we can start to understand the associations between genetic variations and the diseases that they have, the drugs they've taken, etc. And it's an amazing resource, one that's been used around the world. And that means that individuals like myself, I'm of UK heritage, if I take my genetic information and compare it to UK Biobank, I can be pretty sure that any estimates of my risk of disease are going to be very accurate. But if you're of a different ethnicity, there isn't that same set of reference databases to compare to. So I think around the world, it's really important that we start to build up databases of healthy individuals with different ancestries that will allow us to have genomics benefit all of humanity, not just those that historically have been of European descent and the wealthier parts of society. And so Thailand has recognized that. Thailand's got a really interesting kind of melting pot of different ancestries as a country. And so it's starting to gather that data, both in looking at newborns, but also in other basic genomics research and gathering multiple healthy individuals to try and give them the same benefits that we'll likely have here in the UK. And we're involved in those types of projects around the world. We're actively trying to increase the number of non-European genomes that are in the public domain. We just announced in the last couple of weeks a collaboration with Danny Miller in the US, and he's going to help us sequence with our hi-fi non-read sequencing all of the genomes in the 1,000 genome dataset. And it's a legacy dataset now, but one that is a really useful reference because it's intentionally tried to take individuals of lots of different ethnicities from around the world. Doesn't capture maybe everything, but it's a really good starting point, and that's going to again help us understand the genetics of individuals outside of those that are more typically studied.
So we've got the UK, we've got Thailand. What other national governments are moving towards newborn screening? Is this something we're seeing as a trend globally?
It is a global trend. I think every country has some requirements to go through these processes that the UK has done with regards to consultation and where does that data reside and what's the ownership of that data, etc. So I think there are lots of countries around the world that are in pilot stages of doing that, and some more progressed than others. I think world leading at the moment is UAE. So in the Middle East, I think they've had The Emirati Genome Project has been ongoing for 5 years now. They have done a little bit like UK Biobank. They've been typically taking individuals that are healthy, and I think they've sequenced over 900,000 with short reads now, some of those with long reads, to try and assess the genetic variation, link that to the medical records that they have there, such that they're establishing the framework almost of a healthy genome that they can then layer on to newborn genome sequencing onto that. And there's some interesting groups out there. We've got other projects in Greece, I think, as well. There's some small pilot projects in North America. North America is a little bit different from the UK. I think in the UK, there's a relatively easy political argument for doing newborn sequencing at birth because we have with the NHS, we have a single payer system for read a description. That genome will hopefully have multiple uses throughout someone's life course, and the NHS as an organisation can benefit if it makes some cost savings when that individual is 50 because they sequence their genome at birth. Then ultimately there's a return on that investment. Other healthcare systems around the world that are typically insurance based don't quite have the same incentives. So in North America, I'm not sure if this statistic is still true, but on average, it used to be that healthcare was provided as part of your work, part of your employment. And so if someone changed job, which on average people were doing every 4 years, it was not uncommon for their healthcare provider to change. And you can imagine under that situation, if you're healthcare provider A and you've done a newborn genome sequence of that child, the high likelihood is by the time that child has a utility of that genome later in life, that child is no longer under your provision, it's under the provision of one of your competitors. So there's this perverse kind of set of disincentives in the US and in other healthcare systems that are insurance based, but not at a national scale, where I think we're going to see a different rate of adoption of genomics as a whole, actually, not just newborn sequencing.
If we're going to look forward a little bit, if you're a parent or a policymaker listening to this podcast today, what is really important to understand about the potential of this sequencing to improve health outcomes? Maybe not straight away, but in the future, in the life of that child?
Yeah. But every disease has a genetic component. So whether that's, the obvious ones where, for example, a woman may have a very high risk of breast and ovarian cancer because they have truncating mutations in the BRCA1 or BRCA2 genes. At the moment, we don't know that. That young girl is born and they live their life, and then if they're really lucky often in their 40s, they'll have a mammography and someone may or may not find a lump. And that's, it just seems like a really arcane way of doing the screening at this moment in time. If at birth, we could have an understanding what that child's likely risk in their lifetime was of developing breast and ovarian cancer by looking at both those genes, but also at all of the other thousands and thousands of genomic locations that either increase or decrease one's risk of breast and ovarian cancer. It's called a polygenic risk score, can be calculated for that disease and all other diseases. And a healthcare system then has the opportunity to change how we effectively ration screening. I mean, we can't afford to do every test on every person. That's not ever going to be financially viable, and healthcare systems are struggling with the economics of healthcare anyway. But I think the big potential advantage is that we can change the paradigm based on someone's genomic risk score, such that young girl at birth, she doesn't need to know at birth that she's at high risk of breast and ovarian cancer. That's probably not the right thing to do mentally, but at a government level or at a healthcare level, maybe that child is invited to a screen in their late 20s, early 30s because they're higher risk. Maybe it's caught early, the disease is caught earlier and treated earlier, and that individual doesn't die of breast cancer, but is treated very effectively because their disease has been caught early. And yet another young girl may have completely the opposite genetics, be very much protected against breast and ovarian cancer, and they will have the same screening, but maybe they have that in their 50s or 60s. And so you can take the overall budgets that are always limited in healthcare and actually spend more money on the people that need that early detection and spend less on others, and use it more rationally. And I think that's the overall benefit, at least that's the overall potential of genomics. I think we've seen some of those types of studies at small scale bear out. But there's all sorts of interesting ethical and social dilemmas. If you tell someone that they're at a higher risk of obesity, do they exercise more and look after their diet more, or do they just go, oh, and use it as an excuse to not do any of those things? And so around the world, again, I think there's a lot of potential for it to be used for good, but there's a lot of kind of sociology and other types of work that still need to happen to understand how best to take that knowledge that we can gain from genomics and use it to influence people for the better of their health.
Neil, this is certainly just a fascinating area. Genomics is just so fascinating by itself. For you, though, what excites you about what's coming next? Because you've got these big projects in different parts of the world. PacBio is involved in Thailand, the UK, elsewhere. But what's actually exciting you about what's to come, and where do you see PacBio contributing to that?
Well, I think as we touched on right at the beginning, I think the competition between different technology vendors is fantastic. It does drive the innovation much more quickly than it would happen otherwise. So in our R&D team, I'm actually in California today, where most of our R&D occurs. In our R&D team, there's all sorts of exciting new technologies to come that are going to just continue to make our ability to understand the genome, cheaper, faster, more effective. And there are so many areas of humanity it can then touch, and whether that is newborn sequencing or whether that's understanding and predicting the right drugs to take if you unfortunately have cancer, or feeding the world in the future. I mean, we mostly focus on the human aspects of this, but genomics is increasingly being used to better understand how to breed crops so that we can have those that are drought tolerant or understand floods, etc. We've got the ability now to track diseases as they spread around the world, and we saw a bit of that through COVID. And I think increasingly, societies and governments and institutes around the world are putting early surveillance systems in place, because I don't think any of us want a pandemic and a lockdown again like we had previously. So that's a really important part. But it's difficult to single one thing out. And as you can probably tell from the tone of my voice, I get excited about a lot of this stuff. There's some amazing science, and the possibilities of bringing that to bear in a positive way for humanity are immense. And, as a company, we have some ideas of how our technologies are going to be used. But the beautiful thing is you take a set of tools and you put that out into the world, and people come up with, wild and wonderful ideas that you never envisaged in the first place. And so I think we're hoping to see a lot of that as well. Our technology has this beautiful sort of data within there. And yet I think we're still at the early stages of being able to understand the differences at a genetic level and how that really, drives your propensity to disease. We're at this exciting time around the world with large language models and AI. They've done an amazing job of taking the language of English predominantly, but other languages around the world, and understanding that and giving us the tools to better build things in that space. But we're starting to see groups around the world now take DNA, which is just a different type of language. It's the language of life. And if we can understand the language of life and how that fundamentally works with the benefit of AI and large language models, then I think that is going to accelerate our generation of knowledge over the next decade or so. We see little bits of that already with things like AlphaFold, and how Google's team have ultimately developed the ability to take a protein sequence and understand how that folds around on itself and gives the structure and therefore function of a particular protein. And people are taking those tools now and making synthetic proteins, antibodies, new drugs, etc. I feel like a child in a sweet shop. There's so many possibilities ahead of us. It's difficult to pick any one sweet and say that's my favorite, because I think there's just a beautiful set of opportunities for people around the world. And so, if there are listeners that are early in their career stage, I think genomics is going to be an area for many, many years to come that's going to offer a hugely rewarding career, a huge number of opportunities in many different exciting ways. You can take your own personal passion for anywhere in science. I think genomics can help play a role.
Neil Ward, thanks for joining us today on Beyond Biotech.
Absolute pleasure. Lovely to talk to you.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.